Hello. So today we'll be talking about a question that came in GATE 2023 Biotechnology paper. Which of the following vectors are used to clone a DNA fragment of size 220 kb? The options are A. Bacterial artificial chromosome, B. Yeast artificial chromosome, C. Cosmids, and D. PUC19 plasmid. So before going to the answer, we'll discuss a little bit about the concepts of all these vectors. So what is a vector? A vector is basically a substance, usually a piece of DNA, that carries a sequence of DNA or other genetic material and introduces it into a new cell. These vectors act as vehicles to transfer genetic material from one cell to the other for different purposes like multiplying, expressing or isolation. And these vectors are used as a tool in molecular cloning procedures so as to introduce the desired DNA insert into the host cell. The DNA insert that is transmitted via vector is termed as recombinant DNA, and this entire process is very prominently used in molecular biology. Now, these vectors are usually DNA substances that carry different parts involved in different functions. Okay? And these vectors usually have an insert, which is your gene of interest, and a larger sequence that will be the backbone of the vector, which is responsible for the structure of the vector. And these vectors can be classified into different types depending on different characteristics. And the selection of any kind of vector depends on the purpose of the process. So cloning vector is a category of vectors that are essential for cloning procedures, and these vectors have different sequences that enable them to initiate replication in host cells as well as propagate within the host. Okay, so what are the features or characteristics of vectors? These vectors should be capable of replicating autonomously, which in turn depends on the presence of particular sequences in the vector that enables them to initiate replication and propagation within the host cell. Some vectors might even have sequences that allow the production of proteins essential for the inserted DNA, regulation of the process, and further transfer of the insert between different vectors. The size of an ideal vector should also be small enough for it to be incorporated into the host genome. The small size of the vector also enables it to incorporate a large-sized insert for transfer. Vectors should be easy to isolate and purify, as these need to be recovered and reused for multiple purposes. For a vector to be effective, these should also have certain components that facilitate the process of determining whether the host cell has received the vector. Most vectors used in this process have gene that either provides resistance to an antibiotic known as antibiotic resistance gene or produces a particular type of protein. These components are called marker genes. Many vectors also require unique restriction enzyme recognition sites, like here, that enable the insertion of the gene of interest DNA in the presence of specific restriction enzymes. The introduction of vectors into the host cells should be easy, which depends also on a number of factors. And in the case of gene transfer process, it is important that the vector is capable of integrating itself or the recombinant DNA into the genome of the host cell. And it is important that the introduction of recombinant DNA into the vector does not affect the replication cycle of the vector as a whole. So, what are cloning vectors and how are they used? A cloning vector is also a fragment of DNA which is capable of self-replication and stable maintenance inside the host organism. It can be extracted from a virus, plasmid, or cells of a higher organism. Most of the cloning vectors are genetically engineered. It is selected based upon the size and the kind of DNA segment to be cloned. Some of the general characteristics of cloning vectors are that it should be small in size, it must have an origin of replication, it must also be compatible with the host organism, it must possess restriction sites. The introduction of donor fragment must not intervene with the self-replicating property of the cloning vector. A selectable marker, possibly an antibiotic resistance gene, must be present to screen for the recombinant cells, and it should be capable of working under the prokaryotic as well as eukaryotic system. Also, multiple cloning sites should be present. So these cloning vectors are basically used as vehicles for transporting foreign genetic material into another cell, and this foreign segment of DNA is replicated and expressed using the machinery of the host cell. So these are examples of plasmid vectors, and this image right here is a bacteriophage, which is also another type of vehicle or vector for DNA sequence transfer. We'll also talk more about this in the coming slides. So I talked a little bit about all the features of the cloning vectors, how it should have a origin of replication so that it can self-replicate inside the host cell. It should have restriction site for the insertion of DNA and an antibiotic resistance gene for that facilitates screening of the recombinant organism. It should be small. It should be capable of inserting a large segment of DNA and all these. Among these, plasmid vectors were the first vectors used in gene cloning. These are found in bacteria, eukaryotes, and archaea, and these are natural extrachromosomal self-replicating DNA molecules. They have a high copy number generally and possess antibiotic resistance genes. They encode proteins which are necessary for their own replication. PBR322, PUC19, these are some examples of plasmid vectors. So cloning vectors in conclusion are utilized to insert foreign DNA into another cell and create multiple copies of the same, and the foreign DNA is duplicated and expressed utilizing the host cell machinery. So this is how cloning basically occurs. So we'll have a plasmid and we'll have the gene of interest that we want to incorporate within the DNA within the plasmid, and we want to multiply. So the gene of interest we can get amplified using polymerase chain reaction, and then we can cut the gene of interest using a restriction enzyme, particular restriction enzymes. We can simultaneously cut the plasmid with particular restriction enzymes. We can ligate them together and we'll form the recombinant DNA plasmid, and then we can screen for the recombinants using the antibiotic resistance gene, which will be the selectable marker. So in a medium having that particular antibiotic, only cells having plasmid will be able to survive. The rest of the bacterial cells will not be able to survive, and then this recombinant plasmid can be taken forward for various other functions. So the size of plasmids generally range from 1 kb to even 250 kb, and even DNA inserts up to 10 kb can be cloned in the plasmids. Okay. And the plasmids having high copy number is useful for production of greater yield of recombinant plasmid for subsequent experiments, while the lower copy number plasmids are exploited under certain conditions, like the clone gene produces a protein which is toxic to the cells. And plasmids also encode those proteins which are essential for their own replication, and these protein coding genes will be located near the ori or origin of replication. So some of the plasmids are PBR322, PUC19. So what are the advantages of using plasmids as vectors? They're very easy to manipulate and isolate because of small size. They're more stable because of circular configuration. They replicate independent of the host. Their detection is easy because of antibiotic resistance genes. Whereas what are the disadvantages of using plasmids as vectors? Large fragments cannot be cloned, size range is only till 10 kb almost, and standard methods of transformation might be inefficient. So this is how vectors in molecular biology are basically used. Another type of vector is known as cosmid. So what are cosmid? Cosmid vectors are hybrid vectors composed of plasmid and phage vectors. Okay, phage lambda vectors capable of incorporating up to even 42 kb of DNA. So these cosmid vectors are prepared by the insertion of the cos region of the phage vector, okay, into the plasmid vectors. And these cosmid vectors are large size vectors with sizes ranging from 400 base pairs to even 30 kb. And these can carry DNA sequences having sizes ranging from 28 kb to 46 kb. Okay, and these vectors are basically created in order to incorporate large size DNA molecules that cannot be carried by plasmids. And since these are hybrid vectors, these can replicate within the host cell like plasmids or remain packaged like a phage. Now, cosmid vectors do not have many phage characteristics except the signal sequences that promote phage head stuffing. And the hybrid structure of cosmid enables the phage heads to be incorporated within all donor DNA for transfer. The use and production of cosmid vectors have increased over the years as the package system is highly efficient and selective for the recovery of larger hybrids. Okay, so basically the cos sites are necessary for efficient packaging of the DNA into phage particles, and large DNA fragments can be cloned, which is an advantage over plasmids. Okay, and also another advantage might be that they have high transformation efficiency and are capable of producing a large number of clones from a small quantity of DNA. Disadvantage might be that cosmids cannot accept more than 50 kb of the insert. So if your gene of interest is bigger than that, then we might have to look for alternatives. Moving to bacteriophage, which is another prominent vector. So bacteriophages are definitely more efficient than plasmids for cloning DNA inserts. Phage lambda and M13 are very commonly used bacteriophages in gene cloning. Bacteriophages are basically viruses that infect bacteria. That's how the name comes, bacteriophage. Okay, and so these are basically intracellular obligate parasites that multiply inside bacterial cells by making use of some or all of the host enzymes. Okay, and they have a very high significant mechanism for delivering the genome into bacterial cell, and that's how it can be used as a cloning vector to deliver DNA fragments. So most of the bacteriophage genome is non-essential and can be replaced with foreign DNA. And using bacteriophage as a vector, a DNA fragment of size up to 20 kb can also be transformed. Okay, so these vectors can infect bacteria and transform them efficiently while carrying large inserts. Okay, a maximum of around 50 kb DNA can also be packaged into the phage. And so some examples, as I said, will be phage lambda and M13. Okay, so this is how it happens. And in case of phage lambda, it has head, tail, and tail fibers. Its genome will consist of 48.5 kb of DNA and 12 base pairs single-stranded DNA, which comprise of sticky ends at both the terminals. And since these ends are complementary, they are cohesive and could also be referred to as the cos sites. Okay, an infection by phage requires absorption of tail fibers on the cell surface, contraction of the tail, and injection of the DNA inside the cell. Moving to another different kind of vector, bacterial artificial chromosome. So these are very similar to E. coli plasmid vectors. It is obtained from naturally occurring F plasmid, and these are mostly used to study genetic disorders. The advantage is that they can accommodate large DNA sequences without any risk. So these are engineered DNA molecules that are used to clone DNA fragments in bacterial cells, and these consist of a bacteria derived F factor replication origin, which enables the propagation of large DNA fragments in a supercoiled circular form. Okay, and obviously these can carry a very large size of insert DNA as compared to plasmid or phage vectors, and these are considered superior over other artificial chromosomes like yeast artificial chromosomes and mammalian artificial chromosomes, as the F factor found in the bacteria reduces insert chimerism and instability that might arise during the process. And these are highly efficient, as DNA fragments as large as 300 kb can also be inserted into the bacterial artificial chromosomes, which decreases the number of clones and cycles to be performed to obtain the desired result. And these BAC libraries, or bacterial artificial chromosome libraries, have been used to generate large genomic DNA inserts for processes like positional cloning, physical mapping, genome sequencing, etc. And these have been increasingly being used in genetic engineering due to its stability and as compared to other similar vectors. Okay, so the disadvantage might be that the eukaryotic DNA inserts with repetitive sequences might be structurally unstable in bacterial artificial chromosomes, often resulting in deletion or rearrangement. But still, this is also a very prominent choice when your DNA of interest is huge. Moving to the next vector, which is the yeast artificial chromosome, or the YACs. So these are engineered DNA molecules that are used to clone DNA inserts within the yeast cells, particularly Saccharomyces cerevisiae. They have been developed in order to clone even larger sequences of DNA so as to increase the efficiency of the process. So these can clone up to even 500 kb of DNA, which is much higher than the most traditional cloning vectors. And even though these are frequently used as cloning vectors, they are also helpful in other genetic processes like DNA sequencing and analysis. And these are also unique in their ability to clone the complete sequences of larger genomes that exceed the limits of traditional techniques. Now there are some limitations also of using this yeast artificial chromosome, because there's a chance of introducing a high degree of chimerism and insert rearrangement. And also, since these are eukaryotic cells, these are difficult to handle and have lower efficiencies as compared to bacterial artificial chromosomes. And different yeast artificial chromosomes have been created over the years and then used for different purposes. The main advantage is a huge DNA insert can be cloned, and physical maps of large genomes like the human genome can also be constructed. But disadvantage will be that the yield of cloned DNA might be low and the overall transformation efficiency can be low. And also these YACs are not very significantly less stable than bacterial artificial chromosomes. And in YAC clones, there are frequently deletions, insertions, recombination, and other rearrangements within the cloned fragment, and this can actually lead to erroneous interpretation of results when these are used for chromosome mapping. And also, bacterial artificial chromosomes are created faster than yeast artificial chromosomes. So there are a large number of advantages of using bacterial artificial chromosomes over yeast artificial chromosomes, where they're comparatively stable, they're easy to transform, simple purification is required, user-friendly, and also they aid in the development of vaccines. So there are a lot of advantages of bacterial artificial chromosomes over YACs. So coming to our question, the question was which can be suitable for cloning a DNA fragment of size to 20 kb. So obviously PUC19 plasmid cannot accommodate such a huge size of DNA. Even cosmids, even though better than PUC19 in accommodating large size of DNA, even cosmids are not suitable for this size. Only bacterial artificial chromosome and yeast artificial chromosomes are actually suitable for cloning this huge size of DNA fragment. So the answer will be both A and B. Okay. If you're interested to learn more about the different types of vectors used in cloning and learn more about all the types and how these are prepared, how people work with these in the labs, you can consider uh, checking out this course, Genetic Engineering Theory and Application by Professor Vishal Trivedi. There are 3 lectures that talk about cloning vectors, and there are also 3 YouTube videos that have already been uploaded and are in the public domain. So for more information, you can check these videos out. Thank you.